And I'll note that I am somewhat, with some trepidation, taking over from Andy Alspa as director of that course, who's done just an amazing job along with Deb Hogan over the last few years. And looking forward to the challenge and trying to fill those shoes. Thanks so much to Joe and to Melissa for organizing. This is an amazing summit and very unique. I don't think it could really ever happen anywhere else. This coming to Duke to talk about and interact about fungal pathogenesis, fungal infections, is sort of like coming to Mecca. There's very few places. I don't think there's any place else that has such a concentration of imaginative and productive researchers in this area. And so my title changed a little bit. I wanted to highlight something from the previous day, which is fungicidal. We're looking for fungicidal molecules here, as John Perfect highlighted in his first talk. And so a few years ago, we were thinking about how do we find fungicidal molecules, and we came up with this assay, which is we've been using over the last, it's getting close to 10 years now. And it's a very simple assay. Basically all cells on the planet have adenylate kinase in their cytosol. And if you disrupt the plasma membrane integrity, that adenylate kinase will leak out into the extracellular space. And so we reasoned that molecules that cause the cellular membrane to break, either through direct damage to the membrane, messing up the pumps. In other words, if the metabolic machinery stops, then the pumps don't work, things start to leak. And this might be a very sensitive way to identify molecules that were directly fungicidal. And so what happens in our assay is we have something that breaks the cell membrane. Adenylate kinase leaks out into the media. Adenylate kinase then converts ADP to ATP, and then we couple ATP to luciferase generation. And the power of this assay is shown down below here with a couple of graphs. And so caspofungin is sort of the quintessential fungicidal molecule. Disrupts the cell wall, the cell falls apart, stuff leaks out. When you use our assay to find your detection limit, we see statistically significant signal at 12 nanograms per mil. And whereas if you do a standard endpoint growth assay, which has sort of been the de rigueur assay for antifungal drug discovery, you don't see any changes that you can detect until you get to 125 nanograms. So we have a 10-fold improvement in sensitivity for this particular molecule. It doesn't happen to work for all molecules. But for specific ones, we can have much, much greater sensitivity. This is particularly important if you're looking for natural products where you have a mixture of things. You can't really control the concentrations of molecules that you are screening. So what I'm going to tell you about today now is 3 sort of vignettes of how we've used this assay to screen different libraries and have identified 3 different types of molecules which have fungicidal activity. And the first one, so we started out by screening in off-patent drugs. We've heard about this before in yesterday's talks. And basically, this is repurposing. We're looking for things that have already been developed as drugs, and we want to find some that may have antifungal activity. And this also sort of points out our general strategy here for screening. We do initial screen with our library using the AK assay. We do a dose response here as our secondary, make sure that the molecules that we're detecting have a nice smooth dose response. The thing that we don't want to see here is a sort of threshold effect. When you screen, especially for antifungals, you're screening generally at really high drug concentrations, say 50 micromolar or higher. What can happen is your molecules can actually make aggregates. And these aggregates can have activity that the single isolated molecule does not have. And so when you do a dose response, you won't see any activity until you get this aggregation effect. And it's one of the easiest way to remove nuisance compounds. So a dose response is a really important secondary screen for really any any screening assay that you're doing. So we do that and make sure we have a nice sigmoid curve, or at least a nice linear relationship. We then determine the MICs using standard techniques. And because we're looking for things that are basically popping the cell, we do a second assay looking for propydium iodide staining. Propydium iodide gets into cells whose membranes have been disrupted and then stains them, whereas intact cells don't take up propydium iodide. And so we ran through this, through our assay, through the usual sort of FDA-approved Prestwick, Spectrum, whatever library. And I'm going to quickly go through this vignette because we've published most of this recently. Ariel Butts, a graduate student's primary thesis paper, came out about this time last year in MBio. And the molecule that we got really interested in from this screen was Tamoxifen, which is better known as a breast cancer drug. It's used in post-cytotoxic chemotherapy as an estrogen receptor antagonist in maintenance treatment of breast cancers. And it has very good activity against Cryptococcus neoformans with an MIC of 8, not quite as good as against Candida albicans. And this wasn't a new result. People have known about the antifungal activity of Tamoxifen for many years, but really hadn't pursued it. We looked at its interactions with fluconazole, particularly with Cryptococcus neoformans, and found it was synergistic both with amphotericin B and fluconazole, which also sort of got our interest a little further. I'm going to gloss over a whole lot of work here, but one of the most important things that we found was that the drug itself has an MIC around 8. It's well known that these things are converted into a hydroxylated derivative, and then hydroxylated derivative missing a methyl group there, and this is cytochrome P450 mediated. As an estrogen receptor antagonist, these two molecules are actually more potent and more abundant in the patient. So tamoxifen is rapidly metabolized. Thankfully for us, it actually improves the antifungal activity. So we got even more excited here. Usually your metabolism is your enemy, and this time it's actually helping us out a little bit here. So we went, again, glossing over a lot of work here. Turns out that we can show that tamoxifen plus fluconazole in a mouse model of Cryptococcus is better than either of single drugs alone, and gives us about a log plus kill in the brain burden. Now, this experiment is limited by the fact that the metabolism I told you about before is extremely rapid in mice. And so the doses that we're using here provide the equivalent of a 25 milligram dose in a human. Recently, tamoxifen has been repurposed as a treatment for bipolar disorder, and you use 20 times that dose. In humans, you can get up to 500 milligrams into a person, which gives micromolar concentrations in the serum, which then concentrates in the brain to 40 to 100-fold. And so based on what the drug levels we get we know can be achieved in a human brain, we should be 50 to 40-fold above the MIC with this drug. And the further features of this is that it's orally bioavailable. So to summarize, it's fungicidal. We have serum levels. We can get the drug into the brain. I didn't show you the data for it, but it's active against Cryptococcus neoformans within macrophages, which is an important niche for the replication of this organism, and is a place that fluconazole and amphotericin B cannot access. There's no activity of those two drugs against macrophages. And so right now we're as sort of at the stage of thinking about clinical trials for this. It's a safe drug. We could maybe think about putting it together with fluconazole. We'd probably like to do a rabbit study to look at fungicidal activity in CSF, but the more I think about it, I'm not sure we necessarily need that. There's a drug that's already gone to clinical trials, sertraline, which Jerry Wright mentioned earlier. The Boulware group has taken that drug with fluconazole, is doing a clinical trial as we speak. Our drug's actually better than sertraline. And so part of the reason I'm here is to strategize with some of you about where do we go next with this one. And so that's the first drug we came across. The second campaign I'd like to tell you about is we screen a library of kinase inhibitors looking for protein kinase inhibitors that would have it specifically antifungal activity. Now the molecule we came across was one called, at the time, OSU-03012. It's now been renamed AR12. I tried to get rid of all the OSU responses. I trained at the University of Michigan, so I was more than happy to get rid of that. The thing that got us excited about this molecule was it has an MIC of 4 micrograms per mil against Candida. That's what we screened initially. It was in clinical trials and has been through phase 1 clinical trials as a targeted anti-cancer drug. And I'll note that folks who have done that is Arno Pharmaceuticals, and 2 of their members are here at the conference today. So if you're interested further in that, look up Stefan and Alex can tell you about the progress of this drug. It's orally bioavailable, and the serum levels were above MIC in mice, so we were interested in this. We looked at the spectrum of its activity, basically as a 4 microgram per mil MIC against all of the Candida strains that we tested. 4 micrograms per mil against crypto, not as active as against aspergillus, and it has activity against histoplasma, slightly worse than the other pathogenic yeast. So reasonable spectrum. We got some isolates from Ted White that have fluconazole resistance. This one has an elevated MDR. That should be MDR, not MRD. And basically what we got here, it has a 2-fold or a 10-fold increase in fluconazole MIC, and basically all of these resistant strains are equally as susceptible to AR12. And so active against resistant albicans. It's synergistic with neoformans in the presence of fluconazole. So here's a time kill experiment where we use doses of the molecule that are basically fungistatic, OSU here, fluconazole, over time, not much going on, put them together, the bug dies. So that was very exciting. We were happy to see that. And we see the same thing as when we put it together in a mouse. So this is the same experiment that I showed you for tamoxifen. Here's basically no treatment, AR12, fluconazole, and then the combination improves activity dramatically. It's in the 1 log reduction. I'd like to see a little better than that, and I think formulation issues are preventing us from getting better activity. We may have turned the corner on figuring that out, so we may actually have a little bit better activity than I'm showing you here. I pretty much talked about all of this. So this is a reasonable lead compound for sure. We'll see how far it can really go. One of the things that we were really interested in is what is its mechanism of action. And so initially this compound was thought to be a phosphodependent kinase 1 inhibitor. And so we looked to see, that's what got us interested in it, because there's homologs of the human PDK1 in yeast are PKH2, PKH kinases, which are essential kinases in yeast, but are not essential in humans. Turns out OSU does not inhibit PDK1 orthologs as its target in fungi. We purified the enzyme and saw no activity, which was something of a dark day in our lab, since we had a whole grant looking for these inhibitors. We then went looking for targets, initially being guided by what other folks had described in mammalian cells in terms of what the activity of OSU-AR12 was. None of the things that we tested looked to be the target. And so taking a page from Terry's and the Toronto group's approach, we did a chemical-induced haploinsufficiency screen. And we did the entire genome collection of both, so these are heterozygotes of both essential and non-essential genes. And as Terry pointed out yesterday, probably more eloquently, we grow in YPD and then we grow in drug. We use enough drug to reduce growth in the treated part or sample by about 20%, isolate the DNA, sequence it, and basically ask which mutants fell out, which mutants survived, and assayed that by sequencing. And we got about 70 hits out of the whole thing, and from the essential set, we got 5. The 2 that got our attention here, we got ACS2, which is Acetyl-CoA Acetyl-CoA Synthetase. ACC1 is acetyl-CoA carboxylase. Two enzymes in the same pathway in sequence. And when you see two things like that, you pay attention. And so one of the things that acetyl-CoA synthetase does is it modulates the cell's ability to grow on different carbon sources. So we did our sort of metabolomics experiment on the cheap, the old school, what sugar works, what sugar don't. Here's our MIC. This is in albicans. In YP, the MIC goes up a fair bit relative to a standard CLSI, which is in RPMI. That's not very, that's fairly common, so don't get excited about our changes in MIC here. 2% dextrose, we're at 16. Glycerol, 64. One of the things we were originally worried about was this a mitochondrial poison? And this result pretty much rules that one out, because if we saw yesterday that the T2, whatever that drug is, I can't remember the number, that drug, which is targeting the mitochondria, when it was tested in glycerol, the MIC went way, way, way down. And so this is going the other way. So we don't have a mitochondrial poison. Galactose was about the same as dextrose. We were intrigued to see that acetate improved the activity of the drug dramatically. And acetyl-CoA synthetase is required for growth on acetate. Oleate suppresses the activity of the drug, and it does dumping in a ton of pyruvate. And so when we took this together and looked at, okay, let's do our chemotype phenotype matching here, we came across a paper by Mike Lorenz, who's looking at acetyl-CoA synthetase in Candida albicans, which is an essential gene. And when he turned off this gene with a regulatable promoter, cells died on dextrose, they died on acetate, died on ethanol, grew on glycerol, and grew on oleate. So that was very heartening. We were saying, I think we're on to something here. So first thing we did was we got a strain from Mike and asked if we saw haploinsufficiency in the presence of drug in Canada, and we do. We get better killing in the heterozygote than we do with the wild type. So what is acetyl-CoA synthetase? It is right here. It converts acetate to acetyl-CoA. It also, there's two isoforms. One functions in the mitochondria, one functions in the cytosol as well as in the nucleus. Regulates the pool of acetyl-CoA in the cell. Downstream effects of that are sterile biosynthesis, protein acetylation, fatty acid biosynthesis, as well as histone acetylation. So you're talking about a whole lot of very important cellular processes that are dependent on maintaining cellular homeostasis of acetyl-CoA. And so if we were hitting this, it'd be a fairly decent drug target. The reaction it catalyzes, it takes acetate plus ATP, makes this, in the first step, this activated intermediate of AMP, and then CoA comes in and displaces the AMP. So we got some of the enzyme, and what do you know? This now was a very happy day in the lab. Light comes back on, we feel better. We got an IC50 around 5 micrograms per mil. This is the structure of AR12. Our MICs are around 8 to 4, as I said before. That should say 4, sorry. Importantly, we made an analog, dimethyl celecoxib, very similar, has lousy antifungal activity, still inhibits the enzyme, but it's partial and it has a much poorer IC50. We've gone on to make a number of other analogs, and the ones, for the most part, the ones that are good antifungals are good inhibitors of the enzyme, and the ones that are poor antifungals don't inhibit acetyl-CoA synthesis. So we're pretty certain this is at least an important part of the mechanism of action of this set of molecules. We went on to look at a few more cellular phenotypes just to make the sort of the phenotypes and the chemotypes for matching. One of the most important things that the reason we got into this, right, is that the cells lyse, they die. And initially I'm like, how the heck is an acetyl-CoA synthase necessarily causing cells to lyse? And So we tested that experimentally by getting a TET regulatable allele of acetyl-CoA synthetase 2, treating the parental strain with tetracycline does nothing in terms of propydium iodide staining. However, when we shut off ACS2, we basically start lysing our cells, and we also get adenylate kinase release here. So that was a good thing to see. This gene has been, or this enzyme has been studied a lot in Saccharomyces in the setting of aging, replicative aging, because it's in the whole acetyl-CoA NAD sort of pool of enzymes. And one of the things that was reported a few years ago is that if you shut off ACS2, you induce autophagy. And so what we did was looked at ATG8 GFP localization in the presence of drug. And ATG8 is a cytosolic protein that when autophagy is induced, goes to the vacuole and assembles and then is processed as part of the autophagy pathway. And so this increase in staining that we're seeing at sub-inhibitory concentrations of AR12 indicates that autophagy is going on. We have a nice dose response here where we quantitated the number of cells that were staining positive for ATG. A nice response there. And then to further confirm that, we see processing of ATG in the cells that are treated with AR12. So we're inducing autophagy. It's another cellular phenotype that matches our chemotype. And so acetyl-CoA synthase seems kind of fundamental. Is this something we want to be messing up? Are we going to be killing our patient as well as killing our fungi? It turns out we've stumbled, I think, into a really, really interesting, neat hole that has sort of been overlooked to this point. In mammals, acetyl-CoA synthase, acetyl-CoA homeostasis under normal physiologic conditions is regulated by a completely different gene, ATP citrate lyase. It's dependent on citrate, not acetate. We do not live on an acetate-based metabolism. In contrast, yeast do. And so actually acetyl-CoA synthetase has emerged as an interesting anti-cancer therapy or target. And in addition, other targets in the pathway going from acetyl-CoA synthase on to fatty acid biosynthesis, like ACC1, acetyl-CoA carboxylase, have emerged as important targets for regulating lipid biosynthesis in humans and trying to fight obesity and those sorts of things. So we have a target that's essential in yeast, but is not essential in humans. And so we're very excited about this target. We're also excited that, to our knowledge, this is the first structural class of molecules that inhibits this enzyme. There's one report that just came out this month of another structural class, so there have really been no inhibitors of this enzyme up until this point. And so we're excited about doing some more work and trying to further explore this target as well as the molecules we've found. I'm going to finish off this with one final vignette, and this is based on a very large screen. So the first two applications of our assay that I described were very targeted. Maybe the first one was 2,000 molecules, second one was only 100 actually, but specifically chosen molecules that were sort of, the deck was kind of stacked in one direction. We were fortunate enough to have the opportunity to work with the Broad Institute as part of the National Institutes of Health Molecular Probes program. And they took our assay and made it way better. These guys are pros. They know how to screen. And so they got it down to 1536 well format, which I didn't think could have. So we're 2 microliters of a culture of crypto, and we did an effective screen. And so the screen was designed to find cell wall targeted molecules in Cryptococcus. There are very few of those. As we've heard in previous talks in the summit, caspofungin does not work against Cryptococcus, and it inhibits the enzyme beta-glucan synthase very nicely, but it does not have any clinically useful activity. And when you actually look at other molecules that disrupt the cell walls of Ascomycetes, things like alkyl white, Congo red, you have to use much, much higher concentrations of those agents to see phenotypes with Cryptococcus. There's something different about the cell wall. And so we were interested in finding molecules that directly messed up the cell wall of Cryptococcus. And the way we designed this assay is we did our AK assay, and then we quickly basically took out things that were really toxic against human cells. So we screened against 3 human cell lines, and they had to have greater than, the LD50s had to be greater than 50 micromolar. And then we simply asked, does sorbitol suppress the activity of the molecule that we find? Because sorbitol stabilizes plasma membranes of yeast cells that have disrupted cell wall integrity. Sort of acts as a surrogate cell wall. It equalizes the osmotic gradient between the intracellular compartment and the extracellular compartment. So a drug whose activity was decreased by the addition of sorbitol to the media has a reasonable chance of being a cell wall targeted molecule. It's not perfect, but ones that don't certainly are very unlikely to be inhibiting cell wall processes. So that was our secondary screen. And then we had a cell wall integrity reporter where we looked for things that either activated the cell wall reporter or inhibited the ability of calcium white to activate the reporter. And so we carried out, this is what our data looks like, the big mess here, but here's our cutoff, and we had a fair number of molecules. Up here, we actually ended up drawing our line a little higher. The inter-screening Z prime factor for your screening aficionados was pretty good. You want something above 0.5. We were up close to 0.7, so that was excellent. We got only 71 hits, which actually is a decent number. We used a fairly stringent 10 micromolar cutoff. We then found, of that set, 31 were suppressed by sorbitol. We found no molecules that actually activated the cell wall integrity reporter, suggesting we didn't find anything that was disrupting biosynthetic processes that required a compensatory response from the cell wall integrity pathway. What we did find was 3 molecules that inhibited the calcium white induced activation. And they were all from the same class. They looked like this. They were benzimidazoles-thioureas. It turns out it's probably more, this tautomer is probably the right one. It's a benzene sort of thiourea here with an linker to a phenoxy group. And here's the data for the ability to block the cell wall integrity reporter. BRD1 is very, very effective. The MICs for these molecules are okay for hits right out of the screening library with 4 micrograms per mil for our best drug. When we put it together with caspofungin, you would predict that this may have some sort of interaction. And we had a modest interaction. So we had additivity with caspofungin, suggesting that we are indeed interfering with cell wall integrity pathway signaling. And so to test that, here's MPK1, which is the bottom MAP kinase of the cell wall integrity pathway. When you add calcium white, you get phosphorylation of MPK1, as you can see here. When we do the same thing in the presence of our drug, we don't see phosphorylation. So we're inhibiting the activity of this pathway. When we look, we've done a few experiments to say where in the pathway are we inhibiting this. We've looked at PKC1. This is the first essential gene in this pathway. Doesn't inhibit PKC1 activity. So we're somewhere up here. When we knock out MPK1, we still have an active compound, which suggests to me that we're hitting something up at the top, which has probably multiple functions, one of which is to interfere with cell wall integrity signaling. We have done a little bit of an SAR around this, and we know that if we increase hydrophobicity up here, we get a better drug or molecule. If we start to get electron withdrawing groups over here, we see improvement. We know we need this thiourea. We'll get rid of that. The activity essentially goes away. When we look at, compare the MIC versus our ability to inhibit MPK1 phosphorylation, we have a reasonable correlation, not great, but I think we're on to something. Some of our molecules, we've been able to improve the sorbitol suppressive, the difference between the sorbitol and the non-sorbitol up to 10-fold. So we're working our way toward getting a more a molecule that seems to be more specific for the cell wall part of this. I'll remind you that these are completely non-toxic in human cells. We've also done some PK. They're stable in human serum for 24 hours. They're not metabolized by microsomes, so they're a reasonable target as well. These, chemotype. Thioureas have been used in actually a number of different drugs that are already in use. There's some old TB drugs that are thioureas. They get into the brain in general, so we're very excited about this set of compounds as well. And so, some of you know I like to fish, and this is fishing, right? This is what used to be killed in NIH study sections for fishing expeditions. And that's what drug discovery is. It's another, you go fishing and then there's a divine intervention, if you're lucky. The thing about fishing is you got to know where to go fish, and you have to be somewhat smart. If you go fish in a ditch, you aren't going to catch anything. If you go to the Gaspe Peninsula at the right time of the year, you can get some amazing fish. So you got to think about how you want to screen. Every time you go screening, you will get something. You just don't know what you're going to get, and you don't know what it's going to be useful. And the thing you really got to think about is once you got your set, find some assays that will rapidly get what you have in this big net down to what you really want. So that's where the cleverness comes, is how do you quickly figure out what you want. And so hopefully I've shown you that we've been modestly clever and have at least come upon a few interesting leads. So we've shown that tamoxifen as a possible repurposable drug, particularly in combination. We've identified the first-in-class acetyl-CoA Synthetase inhibitor. Appears to us that this may be a fairly decent drug target for further exploitation. As I said before, Arno is working at pursuing some potential antifungal indications for AR12. And we have this new scaffold that we're really interested in looking at, both the mechanism of action, pursuing it further as an antifungal, but also as a probe to try to understand something about how the cell wall integrity pathway works in Cryptococcus. Sorry, I'm going a little over here. Here's some nice acknowledgments. The other thing I'll just point out, Ariel Butts did the tamoxifen stuff. Christy Koselny and Julianne Green did the AR12. We collaborated with the Broad Institute, as I mentioned. Whole bunch of folks up here. Joe Heitman has been our collaborator who sort of got us into the whole Cryptococcus world. If you don't like to work with people, go home. Because drug discovery is you can't work on drug discovery alone. You've got to have friends and you've got to work with them. This is not the field for an autistic individual. Thank you.